CODIT stands for compartmentalization of decay in trees. And CODIT was a concept that was developed by Alex Shigo. Dr. Shigo was a U.S. Forest Service scientist who was really a pioneer in the study of decay in wood. He sort of came of age after the development of single-person chainsaws, and he went to town. He went into the woods, he was a pathologist, but he went into the woods and made innumerable, thousands and thousands of longitudinal sections of trees and studied the patterns of fungal decay and how that fungal decay was contained over time by these trees. Shigo obviously was a very hands-on person, and actually one of his sayings was touch trees. So I think many of us can appreciate that learning and science often happens best when we engage, with whatever it is we're learning about or studying. And so Alex Shigo, real pioneer in this area, and again, developed in I believe in the 60s, 70s, maybe even into the 80s, but these concepts are still valid and sort of being built on today in terms of a nice framework for understanding how fungal decay is compartmentalized, but this actually also applies in some ways to other pathogens in addition to fungi. So we'll go through the CODIT concept, which really involves 4 different walls or barriers. And the starting point for understanding CODIT is the idea that trees are already highly compartmentalized organisms. When you look at xylem in trees, you can actually already picture, boxes. So this is a cross section of a piece of angiosperm wood, and so a growth ring goes from here to here. And what you can see is that, for instance, we already have boxes that separate different compartments of vessel elements, which are one of the things that are being protected in defense responsive trees. And those boxes are formed by the growth ring barriers and the rays as barriers. So Shigo said these barriers are already there. This was a model that he showed where, here are rays and the growth rings, and they're forming a literal box around these vessel elements and fibers. So Shigo said, trees are already compartmentalized, and some of those existing walls already form barriers to the progression or movement of a pathogen through the stem of a tree. So he just formalized some of these existing compartments into walls 1, 2, 3, and 4. So this diagram sort of lays out at least 3 of those 4 barriers in terms of walls. So Shigo said wall 1 is an induced response when vessel elements or tracheids are plugged and prevent the vertical or longitudinal movement of a pathogen through the tree. So he said the top and bottom of that compartment forms when the vertical elements are plugged after wounding. So wall 1 can be considered an induced response because that usually doesn't happen until after wounding. So one way that those vertical elements become plugged is by the formation of tyloses in angiosperm vessels. So these are examples of the formation of tyloses in angiosperm vessels. Here we're seeing these vessel elements, and this is the formation of tyloses that are formed basically by ingrowth from ray parenchyma into vessels. And that eventually clogs or blocks the entire vessel element and forms a plug. So these are examples here. This is a cross section. This is a longitudinal section. This is a cross section showing tyloses, in this case in white oak. And white oak is a classic tylosis forming species, but those are formed by ingrowth from ray parenchyma into the vessels, and that plugs those vessels and blocks vertical or longitudinal movement of pathogens. Now, this wall 1 can also form those plugs from resin plugs as well. And sometimes gums, resin or gum plugs can also form wall 1. Tyloses are an active biological growth into the vessel elements, while resin or gums, resins would be probably more specific to gymnosperms, where resins are exuded into tracheids and serve as a blockage. Gums are another chemical that may be produced in other angiosperms that can also be used to form wall 1. Now, wall 2 and wall 3 are constitutive walls that are already there. So wall 2 is the growth ring, the former growth ring. So in thinking about this compartment, the growth ring, the previous growth ring where it ended, is a barrier to pathogen movement towards the center of the stem. So there's some resistance or barrier to movement through those final fiber, tracheids, or vessel elements that are formed at the end of a growth ring, and that forms some barrier to inward movement of decay. Wall 3 is formed by the ray parenchyma, and that would prevent movement of pathogens in sort of a circumferential pattern. So prevents movement in that direction in the stem. And then wall 4 is not on this diagram, but basically if we're thinking about this compartment here, wall 4 is the subsequent growth ring. And that prevents movement into new wood or new xylem that's formed after the attack. Here's an illustration of compartmentalization that occurs due to the CODIT response. So there was a wound on this tree, a branch broke off, or there was some other wound, and the barrier walls sort of contained the spread of that decay. And the next diagram I'm going to show you, this is a longitudinal section through that stem. And the next cross section I'll show you are from this cross section where there's section A in an uninfected or unwounded part of the tree and the infected part of the tree. So in that uninfected part of the tree, I've already showed you this diagram. These are just the preformed compartments. But in that infected part of the tree, you can see that wall 4 is the formation of the subsequent growth ring. Okay? So that subsequent growth ring formed after the wounding event or after the pathogen attack. And what you can see is that in addition to the sort of natural mechanical barrier that occurs at growth rings, there also is additional deposition of, I would say, depending on the species, these are likely polyphenols or resins or gums, and they're probably also additionally lignified. Okay, so this is an induced response. So wall 1 and wall 4 are induced responses. And that wall 4 provides a pretty strong barrier to movement of decay. Well, Shigo actually ranked those walls from weakest to strongest in terms of their resistance to decay movement. And he said wall 1 is the weakest. Okay? And you can sort of see that in the spread of decay, where this wound here allowed decay to spread quite a ways longitudinally. Okay, so that resistance caused by plugging the vertical elements did eventually stop the spread, but it allowed more spread than was allowed radially. Okay, so that wall 1 is the weakest response. Wall 2 and 3, the previous growth rings and the rays are constitutive. They're already there. They're the next strongest. And they help to prevent spread. So you can see the wound actually happened right here. So at this growth ring. Okay, so you can see the wound spread inward into previous growth rings. So there was some resistance, but it was not as strong as, for instance, the wall 4, which occurred right here in the growth rings after that wound. So what that means is that wall 4, it's the strongest. You seldom see spread of decay, fungi, into new wood formed after the wound. Sorry about my handwriting. So that barrier, that wall 4, is that new growth ring formed after the wound. And you can see there is no spread of decay into that new wood formed after that wound. So that prevents spread of decay into the new wood. And with the formation of that wound periderm and the inward curving of the vascular cambium, that also gradually closes that wound. So together those form sort of a coordinated response that compartmentalizes that initial wound and the spread of decay. This image that I got from Andrew Kozer really nicely illustrates that. It looks a lot like Shigo's illustration, where there was this wound and the wall 1, wall 2 and 3, and then wall 4 basically contained the decay in that magnolia to this sort of compartmentalized zone. So it's often said that trees don't heal wounds, they compartmentalize wounds. So they really can't do anything to fix the decay that happens, but that CODIT response, both constitutive and induced, helps to limit the spread of that decay and compartmentalizes it and allows the tree to grow around it to grow new wood. As you can imagine, that compartmentalization, as I mentioned earlier in the lecture, is one of the things that allows great longevity in trees. If this decay was allowed to spread unchecked into new wood, that tree would be doomed and it would eventually lose structural integrity. But that ability to compartmentalize and stop the spread of that fungi allows that tree to continue growing, continue getting bigger, and to live a long time, even after wounding or decay. And that's one of the sort of unique and wonderful things about trees and their response to wounding and pathogens. As with all things related to physiology and responses, there's genetic variation in how trees respond. And one of the ways that manifests is differences in how well different species compartmentalize decay. So this is a table from an extension document that sort of categorizes some different species, in some cases different genuses, in terms of how well they compartmentalize decay. You can almost look at this as a division in how well different species or genuses of trees do CODIT, where these trees, they all do some CODIT to some degree, but these don't do it very well. There's more spread of decay in these trees than there might be in these species of trees. So this is really interesting and useful information to have for management, especially, for instance, in management of trees in the landscape. You know, if a walnut tree is wounded, we know that walnut compartmentalizes decay pretty well and is likely to be okay. Whereas, for instance, if we have a poplar tree in the landscape that's wounded, we know that it may not compartmentalize that wound very well and may have problems, structural problems, down the road. So this is useful information to know with regard to genetic variation in compartmentalization. Okay, so some trees do CODIT better than other trees do. The final thing that I want to cover has to do with phenology. So phenology can be thought of as sort of another element that's related to susceptibility to disease, in that phenology can influence how resistant or not a tree is to attack by a pathogen. One example in the literature is that Norway spruce becomes progressively less resistant to fungi carried by the spruce bark beetle in spring and summer. So Norway spruce is more susceptible to that fungus in the spring and summer than it is in the fall and winter. That's often has to do with the availability of rapidly growing tissue that may be more palatable to or vulnerable to that fungus. These pictures show fusiform rust fungus attacking or that have already attacked southern pine. So these galls on the branches are examples of fusiform rust galls on trees. Fusiform rust is a fungal pathogen of southern pines, and this sort of bushy, multi-stemmed form is an extreme response that we often see in extreme fusiform rust attacks. And that kind of attack is manifested under intensive management. So we see more extreme fusiform rust in more intensively managed southern pine plantations, partially because that intensive management creates larger amounts of green succulent tissue that is then more susceptible to attack by the fusiform rust fungus. And that's an example of a phenological effect where that greater availability of succulent tissue that is more susceptible to attack by the rust fungus is a phenological response that's caused or induced by that intensive management. So certainly we can modify the phenology of trees and make them more, or in some cases less, vulnerable to attack by fungi. So that's another important element that influences the susceptibility of trees to attack and part of their defense or lack thereof.